The replication of the E. coli chromosome begins at its single replication origin. Both strands are synthesized simultaneously by a dimer of DNA polymerase III holoenzymes, which together with the primosome form an assembly called the replisome. Here we will step through the synthetic process at a replication fork. All DNA polymerases synthesize DNA in a 5' to 3' direction. That is, they attach incoming nucleotides to the 3' end of the growing daughter strand. They require a single-stranded template, nucleoside triphosphates, and that the strand they are extending form a double helix with the template strand. The tricky part for DNA replication is that although the replisome must move along the chromosome in a single direction, it must copy the DNA strands that it encounters in both the 5' to 3' and 3' to 5' directions. Therefore, one strand has to loop around in order to come back through the second polymerase in the correct direction. In order to provide single-stranded templates for the DNA polymerase to copy, DNA B protein uses the free energy released by ATP hydrolysis to unwind the DNA helix in advance of the replisome. Because of this helix unwinding function, DNA B and related proteins are called helicases. Single-strand binding protein then coats the newly unwound DNA strands to prevent them from re-annealing. DNA polymerase III now has single-stranded DNA to copy. Each holoenzyme unit copies a strand of DNA, synthesizing the new strand in the 5' to 3' direction. Leading strand synthesis is always ahead of lagging strand synthesis, because the lagging strand must loop around to enable the DNA polymerase to synthesize in the 5' to 3' direction. The lagging strand is synthesized in segments, called Okazaki fragments, which are eventually joined by DNA ligase. The leading strand is synthesized continuously. When the DNA polymerase III holoenzyme copying the lagging strand encounters the previously synthesized Okazaki fragment, it releases its bound template strand. The synthesis of the next Okazaki fragment is initiated by the synthesis of a short segment of RNA that is periodically inserted by a protein assembly known as the primosome. Note that the primosome must reverse its direction of travel when it synthesizes the RNA primer. After the additional lagging strand template is unwound, this RNA primer will find its way to the empty holoenzyme, which then extends the strand to yield the next Okazaki fragment. The lagging strand now contains a series of RNA primers that must be removed. DNA polymerase I uses Nick translation to replace this RNA with DNA. The DNA strands are then joined by DNA ligase. As the replisome continues to move along the chromosome, successive Okazaki fragments are synthesized and joined to keep up with the synthesis of the leading strand. Having completed one Okazaki fragment cycle, we have now returned to where this story started, albeit a bit further along the DNA.